Let's look at this quite complicated question on estimating the number of colonies that you'd expect to develop on an agar plate. The mathematical component here is looking at serial dilutions and using that information to work out the number of bacterial colonies. So reading the question, serial dilutions can be used to estimate the size of a bacterial population in a culture. A scientist used 20 centimetres cubed of a bacterial culture that contained 1 times 10 to the power of 6 bacterial cells. 5% of the 20 centimetres cubed culture was transferred into a new test tube and made up to 10 centimetre cubed with water. An additional tenfold dilution was carried out, which produced a final 10 centimetre cubed solution. 0.1 centimetre cubed of that final 10 centimetre cubed solution was transferred onto an agar plate. Each colony that developed on the agar plate was assumed to represent a single bacterial cell in the bacterial culture. Estimate the number of colonies that you would expect to develop on the agar plate. So there is a lot going on in this question, and there are lots of different mathematical skills that are needed as well. So if I was going to have this question, I would be drawing it out and understanding the process a bit better. So I'm going to start with here 20 centimetres cubed of a bacterial solution. So my original solution here in a beaker, let's put something in there, 20 centimetres cubed. And of that, in there we have got 1 times 10 to the power of 6 bacterial cells. So that is 20 centimetres cubed. We've got 1 times 10 to the power of 6 bacterial cells. To make that a bit easier, I'm just going to change that into a million because that's just an easier number to work with. So it's got 1 million bacterial cells, and that's what that means there. Okay, the next thing that happens then is that 5% of that gets transferred into a test tube. 5%. So it gets transferred into the test tube and then topped up with water to get to 10 centimetres cubed. So 5% of a million then, we can work that out by doing 1 million times by 0.05. And that answer is 50,000. So we have now got 50,000 bacteria in test tube number 1. The next thing that happens is it says we're doing another tenfold dilution here. So again, we're going to take 1 centimetre cubed of this because we're doing a tenfold one. We're taking 1 centimetre cubed and we're going to top it up with 9 centimetres cubed of water. So we're taking 10% of this solution into this one. So there we go, 1 centimetre cubed gone in and 9 centimetres cubed of water. So because it's 10%, we need to then times 50,000 by 0.1 to get our 10% value, which is 5,000. So that's how many bacteria are in that second test tube along. And then the next part is it gets put onto an agar plate. So let's just have a look here where we're 0.1% goes onto an agar plate. Now if I've got 10 centimetres cubed, 0.1, not percent, sorry, centimetres cubed, that is 1%. So I'm going to do 5,000 times by 0.01, and that is going to give us 50, and that is what's going to be transferred onto the agar plate here, and those bacteria are going to be allowed to develop. So the number of colonies in that agar plate is 50 for the 3 marks.